For those of you who are on already, as you know, this is Kristin Joivell. We're really excited to have her be doing our fourth Teacher with Alum webinar. This one is open to other teachers as well. So we'll do a brief intro and then let Kristin go through her presentation and leave 10 or so minutes at the end for Q&A. If you have any questions for Kristin, there's a chat box that you should be able to see on the right side, so you can just type your questions in there and we'll answer them at the end. And Kristin will also be following up later, so if you want to reach out to her individually later on as well, she'll be available for that. So as you all saw in my earlier messages, Kristin was a senior fellow, so in the past few years we started having senior fellows do workshops on our teacher teams, all about bringing it back into the classroom. So she was a senior fellow in 2016 on our expedition Climate Change at the Arctic's Edge in Manitoba in Churchill in Canada, and she also was a fellow in 2010 on our expedition Coastal Ecology of the Bahamas. So during this evening's chat, she'll be sharing how she organized and developed a pond project for her students in their after-school program, and it culminated in a family science night. So really excited to have Kristin here to share experiences. Go ahead.
So welcome everyone. Just to give you kind of a point of focus of where I was coming from developing this project, I just wanted to show you a few things about climate change at the Arctic's Edge, just to give you a frame of reference of where I was coming from creating the project. So I went up to Churchill, Manitoba with the Earthwatch Institute as a senior fellow in summer 2016. And of course it was in Churchill, Manitoba, which is up in the Hudson Bay lowlands area. So that's up in northern Canada, not quite at the Arctic, but on the Arctic's Edge as in the description. As you can see here from the airplane, the Hudson Bay lowlands area is basically a whole bunch of pools everywhere teeming with life, which is surprising because it gets so cold up there, especially in the winter. So you can see the ponds are everywhere and all teeming with life. And we certainly found out a lot about that while we were up on the expedition. You can see there's all kinds of different kinds of ponds. There's larger ones, smaller ones, coastal ponds. And the scientist in charge, Leanne Fishback, had a lot of different studies going on throughout the area. The studies were ran through the Churchill Northern Study Center, which is out in the field. It's a really great field campsite where you can actually live in the Northern Study Center while you're studying and going back and forth from site to site. So because of the location of the area, you can't camp out in the field because of polar bears and that kind of thing. So they have this great Northern Study Center where you can work out of as your base camp. And here's our head scientist, Dr. Leanne Fishback, who is the coordinator up there at Northern Study Center. And of course, as part of the expedition, we had to gear up each day to go into the field from the study center because of all the mosquitoes. As you saw in the photographs, you know, it's basically a bunch of ponds all up in a wet area. So of course, there were thousands and thousands and thousands of mosquitoes. So each day we had to gear up in the nets and the different equipment to keep the mosquitoes off of us. We couldn't use any repellent because of the ponds and the sensitive life in the ponds. So because of that, we had to be covered up at all times. Also, we had armed guards protecting us at the ponds because of the polar bear threat in the area. If you didn't know, Churchill is the polar bear capital of the world. So if you're interested in seeing polar bears, active polar bears, you can go up there and see them during the right season of the year. They have tundra buggies and all kinds of neat ways that you can safely see the polar bears and interact with them in a safe way. And this is how we traveled to collect data each day in the ponds. We had a Northern Study Center van that we traveled around the tundra in. And as you can see here, we're ready to collect our data and we're all geared up. Lots of gear. So we're starting into the field and you can see we have our waders on, we're protected from the mosquitoes, and we have all of our equipment. And these are the research questions that Dr. Leanne Fishback was helping us figure out as we were studying up in Churchill. We were there for about 2.5 weeks in the ponds. So you can see we were studying a lot of questions, mainly focused on growth rate, species assemblage, and palm chemistry. And then of course, looking at the environmental impacts of climate change on the Hudson Bay lowland wetlands area specifically. before we started our research and before we started working in the ponds, we did have to kind of get studied up on all of the different organisms that lived in the pond. So, you know, we were looking at amphibians, we were looking at fish, and then looking at macroinvertebrates, as you can see in the bottom section there, all kinds of animals that don't have backbones. To figure out which was which, we had to use the identification guides that Dr. Fishback had designed for us to use in the field. She works with many Earthwatch teams throughout the season. So of course she has a lot of resources to help people who aren't professional scientists know how to collect data in the field accurately and systematically. And I was really interested in that because I was trying to figure out how to do a project with students who, you know, aren't professional scientists, trying to figure out, you know, how I could help them mitigate all this information that we were going to be seeing on our project. So trying to figure out, based on what Dr. Fishback did, the ways that we could collect data in our ponds here in Pennsylvania as well. So you can see there was all kinds of specific things we were looking at with identifying the fish and amphibians, and also identifying all of the macroinvertebrates, the creatures that didn't have the backbones. You can see there was a huge variety we were looking at and documenting throughout the experience. And some of these I had heard of before and seen in Pennsylvania, and some of them I had never heard of before. So every day in the ponds, we had 5 different tasks that had to be completed by different teams, and we rotated through the teams throughout our experience, so we did get to do all of the jobs. For physical data, we basically measured the pond using a GPS, walking the perimeter of the pond, measuring depths in the pond as well. For environmental data, we used a sensor in the pond to look at the water quality. And as you can see, there's a lot of numbers that go along with water quality. It's not just clear or cloudy. There's all kinds of numbers with dissolved oxygen and turbidity and all kinds of information about the water health. And then we also collected water samples and ran them through a vacuum pump to collect the sediment that was in the ponds. Also, we used traps. These are just recycled soda bottles that we used, and the grad assistants set them in the ponds the night before for a certain number of hours. And then the next day we would look in the traps and see what we found. We would also find wood frogs, which are one of the types of frogs in the Hudson Bay lowlands area. And of course we'd find the tadpoles of the wood frogs, and then also tadpoles of the boreal porous frog. And here's an adult. And then one that's probably just metamorphed into an adult, and a very young one. You can see it's even smaller than a fingernail. So these are the kind of things we were finding in the traps, amphibian-wise. We'd also find predaceous diving beetles, both the adults and the larvae as well. And then in addition to traps, we did some dip netting. So basically taking a net and going around the pond and just dip netting for specific periods of time in the pond. And then taking the material from the net and sorting it, and then identifying based on presence or absence of the creatures we were looking for. With growth rate, we were mainly looking at the growth rate of tadpoles. So we would capture a certain amount of tadpoles within a pond if possible, take them back to the lab, and then weigh them on a very, very accurate balance scale. It was really hard because some of the tadpoles weighed less than 1/1000th of a gram. So, you know, you have to really be careful with the tadpoles and make sure you don't injure them, because the next day because as soon as we're done weighing them, they go right back out to the pond, the same place where we found them, so that we're not destroying any life in the ponds. So I did want to show you a little video here of just how it looks when we're collecting data. And then I'll show you the statistics here. So let me get that on here for you. Okay, I think you can see it. Let's see. I don't know if you can see that. Can you see that?
No, but it doesn't look like it's up yet on our end.
Let me share it again here. Oh, here we go. How about now?
Yes, perfect.
Okay. So this is just a speeded up time-lapse video that someone recorded with a GoPro while we were collecting the data in just one of the ponds. This is kind of what it looks like doing all 5 of those jobs in action. You can see everyone's very busy collecting all of the data. So when we were doing this, I was kind of envisioning, you know, how could this look with students, kind of envisioning like the problems that could happen, and envisioning, you know, how exactly I could get students organized enough to be able to do jobs in the field and not damage the environment, and also be able to find out some cool science information. So hopefully hopefully that gives you an idea of, you know, where I'm coming from with trying to think of this idea. So now I'll pull up this. Hopefully it will share this for me. And I'm kind of new at this technology, so bear with me. Okay, I think I have it now. Can you see that?
Yep, perfect.
Okay, here we go. Okay, so now I just kind of gave you, again, a little idea of what I did up in Churchill with the team, and just kind of gave you an idea of where I was coming from, you know, collecting the data in the ponds. And so when I got back to school, I was already kind of thinking of ideas of, you know, how can I bring this to the students and how can we develop a project from this? So basically I developed the Juniata Valley Pond Project, the JV Pond Project, for our elementary science club. I decided to do it as an after-school program. Of course, some of you, I'm sure, are teachers, and I'm sure you know how hard it is to fit in extras, you know, even with all the great curriculum we're doing already, and then trying to do a giant project on top of that, it can be very difficult. So since I have an elementary science club already, and I'm one of the advisors for that club, I decided to do it as an after-school program. One of the weaknesses of that is that not all the students will be able to participate because it's hard getting rides, you know, sometimes for some students. But also one of the benefits then is that we have the time to be able to use as much time as necessary after school to do the job that we want to do. So there's benefits and weaknesses to the after-school program. So of course the idea for this came from Churchill, and like I said, when I was up there working, I was kind of trying to think of how it would look doing this with children, you know. I mean, obviously we're all up there, we're adults, and the children that I wanted to work with were grades kindergarten through 6th grade. So I wasn't just going to be working with 5th and 6th graders or high school students. I was going to be working with children ranging in age from 5 to 12. So I wanted to try to think of ways that I can involve all the children, even the younger students. So I thought that one of the ways that the children could collect data would be doing dip netting. That seemed to be a really easy way that you could mitigate the ponds without causing much damage. And then also figuring out the presence and absence of species. So not really worrying so much about counting specific numbers of species, but just trying to figure out whether they're there or whether they're not there in the pond. So I thought that that would be a good way to go with elementary students. Of course, with high school students and older students, there's lots of other ways that you could do it that are more involved. But with the age group I was working with, especially it being an after-school program, I decided to do it that way. So our ponds looked like this in February. So basically, to get this started, I tried to find some community partners, you know, and here we're talking about developing community partnerships through doing science research. So one of the first things I needed to do was find a community partner. And I had interacted with some of the scientists in PARS, which is the Pennsylvania Amphibian and Reptile Survey team, through my work with the Audubon Society. I had been the field trip coordinator for a few years, and we had done some field trips into some vernal pools. And I thought maybe the PARS team would be able to help me. So I contacted our PARS representative, and he was willing to come out and help collect data in ponds. So we drove around Huntington County looking at different ponds and trying to think, you know, how are we going to mitigate these with a school bus? How are we going to get the school bus parked or a school van? You know, is it safe to go to these ponds for children? You know, so kind of looking at all those things. So these are what the ponds look like way back in February 2016. You can see there's snow and ice on the ground. You know, some of them are larger, some of them are smaller. And, you know, I'm sure if you work with children, you're already thinking, you know, different things that could happen at these ponds with different logs and things to fall over. And, you know, sometimes there was deeper water and shallow water. And of course, the gear the children would need to be able to explore these ponds. So those are kinds of all the things I was thinking about in January and February when the PARS rep and I drove around and kind of looked at ponds. And then from there, he and I, depending on what he knew already about species assemblage at the ponds and what I knew about children being able to mitigate certain areas safely, we kind of picked about 12 ponds that we were going to be visiting throughout the study. So then we were just kind of waiting for the spring happenings to start occurring. So while we were waiting until, you know, March for this to start happening, I needed to start thinking of ways that the children could be able to collect this data once we got them out there. So I decided to make identification guides similar to the ones that Dr. Fishback used up in Churchill. So we were going to be looking for frogs and toads. And I made these identification guides very simplistic, based on what the PARS representative was talking about in the data that was on the PARS website. And PARS is like a citizen science website. So all kinds of data go there from lots of different places in Huntingdon County here in Pennsylvania, but also all over the whole state of Pennsylvania. So talking to our representative and figuring out what toads and frogs we might be able to see, or we probably would be likely to see in these areas, is how I decided which species. And I thought working with K through 6 students, kindergarten through 6th grade students, probably about 6 species was probably the maximum amount we wanted to have. And as you can see, I did the identification guides as photo guides and kept the simplistic common name there, but also had the scientific name there too for children to look at depending on the age. So you can see we also were looking for salamanders and newts. You can see here the ones that we probably were going to see in the ponds that we selected. And then looking for turtles. And here's the ones on our identification guide we selected. And then I'm sure you're probably wondering, if you're not familiar with ponds, what this crazy thing is. And I didn't even know what this thing was until a couple years ago. So this is a fairy shrimp, and that's its eggs there at its tail end. And so these live in ponds actually all over Pennsylvania, and they actually live in ponds in Churchill. And when I was in Greenland this past summer, they live in ponds in Greenland, right up near the ice cap. So they're all over the place. You'll have to check your local ponds and see if you can find some fairy shrimp. But they're a really neat kind of animal. And they're one of the macroinvertebrates we were looking for. So you can see them here on the identification guide. And instead of doing sketches and things like that, I stuck with photographs because children, you know, being able to look at organism and then look at a photograph, I think it's a lot easier for children to match that up. Maybe we have a little bit of error, you know, and maybe not as accurate as sketches and with all of the data points on them. But for young children, I thought that this would be a really good way to go. We also took a look at plants with the wetland plants, because of course a lot of the organisms are feeding on the plants and laying eggs on the plants. So I thought that would be important information to look at too. So here was our schedule, and this, I didn't regret it later, but it was really a hard schedule. But the children did really well with it. You can see, of course, this was a schedule right after a school day. So dismissal is at about 2:45. And then I gave them a little break right after dismissal and provided a snack for them so they could kind of rest a little bit. Then after that, I did a little presentation just to kind of give them an idea of what we're going to be doing, you know, what is the project all about? Because for each visit to the ponds, which we did 8, there were different children. To get the kids interested in this, I did a whole bunch of announcements on our PA system, sent a lot of things home in the newsletters for parents, and, you know, just talking to kids in the hall and getting them excited about it. And I was planning on 6 trips because that's how many days the van was available for that I could find between the beginning of March and mid-April, which is when most of the action is happening in the ponds with egg laying and breeding. But so many kids signed up that I actually had to do 8 different dates so that I could accommodate all the children, because they didn't want any kid to be turned away. So total, I had about 48 children coming to the ponds total, with 6 of them coming each time, since the van could hold 6 children at a time. And 6 is really a good number. You don't want too many kids stomping around in a pond. It'll destroy the area and mess up the eggs and kinds of things like that. So it actually worked out really well that so many kids signed up, but that the numbers were limited naturally by just 6 being able to fit in the van. You can see there from 3:20 to 3:40, we spent about 20 minutes each day disinfecting our gear before we even left the school. There's certain disinfection methods used by scientists in ponds. The PARS representative taught me about how to disinfect the gear. So I went through gear disinfection every day before we traveled even to the first pond site. That way any of the kids' waders or rain boots or anything like that that might have any viruses on them from other ponds or other sites, those would be disinfected before we even left the school. We'd travel to the first pond, do about 2.5 hours work in ponds. Usually we would visit 2 sites per day. So we'd work in the first pond usually for about an hour, travel to the next pond for about 20 minutes or half an hour, and then work in the second pond for about an hour also. And of course we'd disinfect in between the ponds as well. So when we were getting ready to leave the first pond site and then we'd get to the second pond site, before we got into the second pond, we would disinfect our gear again another time, just to, you know, control the virus spread. There's a lot of viruses that affect amphibians in ponds in Pennsylvania, so we did need to be very careful about that. Then, of course, taking some time to travel back to the school, another disinfection, because we don't want to take the germs that are in the ponds we're studying home with the students on their gear, and then eating dinner together. And then after eating dinner together, I thought a good way to incorporate the children's experience that they could respond to it would be to do some art. So each night I picked a different media like watercolor or chalk or a tempera paint, and just gave the children, after they were done eating, about half an hour or so to respond to their experience through art. And you'll see some of the art later in the presentation, but they really enjoyed responding to the pond that way. I think it was really neat to see the different ages of children and how they either focused on a certain species, or they drew a picture of what they were doing in the pond, or they focused on the whole pond. So you'll see some of their art later. But that was the schedule. And as you can see, you know, it's 5 hours of craziness after a full day of school. But I mean, I had 5-year-olds there, I had 12-year-olds there, usually a mix of ages each night. And it really went surprisingly well. Like, the children did a really great job holding in there and hanging in there for 5 hours of work. You know, and it was kind of like working in the ponds in Churchill. You know, you had to really buckle down and focus, you know, to be able to do science research for so long. So this is how we recorded the data. You know, we had those photographic identification guides, and this was one of the handouts that was emailed to you. And also I did have those photographic identification guides emailed to you too, just in case you're interested in seeing those more up close. So you can see this is what our data sheet looked like. And what we'd do is, after we would collect all the data in the first pond, we'd sit in the van and kind of have a snack in between the ponds, and we'd fill this out together as a group. So you can see here's a completed one for a pond on April 5th. You can see we found no turtles or nothing specific with plants that day, a few extra plants that weren't on our list, but we were finding some macroinvertebrates and some frogs and toads and some salamanders. And then, of course, some signs of other animals too, including garter snakes and raccoons, scat. So the children were excited to find any evidence of any animals possible. So protecting the ponds is really important, you know, not just with the disinfecting, but also with having, you know, 6 children stomping through a pond. You don't want them destroying egg masses. You don't want them wiping out an entire population of salamanders or frogs from one pond visit. So another thing we were doing besides getting in the water at the ponds was that we were rolling over logs to see what was underneath, because a lot of the salamanders will hang out under the logs. So in the logs around the ponds, I would teach the children how to roll the log backwards, look underneath, move any organisms out of the way, and then roll the log back over again, and then let the organisms find their way back under the log. The big thing not to do, and I didn't realize this till I started working with PARs, is to roll the log over and then roll it back over on top of a creature. You don't want to do that. They can get trapped under there. It's really sad when that happens. So there's certain ways that I taught the children how to do that. And then, of course, the other thing is always putting the log back when you're done, even if you don't find organisms under it. It's important to put the log back because somebody might not be at home when you roll the log over, and they might want to go home again to their habitat or their burrow. And if you leave the log rolled over, they're not going to have any place to go back to. So, you know, just really working to protect the habitat. Of course, trying not to be stomping through the ponds. You can see here, this visit was at the beginning of March. So, you know, it's very cold out. You know, we've got on winter coats, earmuffs. It's really chilly. And there might even still be some frost and ice on the ponds. But we were still finding life even at the beginning of March, especially the wood frogs that were coming out very early in 2016. So you can see this little girl here. She's, you know, mitigating her way through the pond so that she's not stomping through all of the egg masses and different things that the wood frogs are doing underneath her there. And of course, here's our disinfection process. You know, using what we're using is like about a cup of bleach, a quarter cup of bleach to a gallon of water, and that's basically the ratio. And you basically just splash all your gear and make sure it's rinsed before you enter the pond. And then, of course, rinsing all of the boots before we go to the pond too. So using that bleach and water solution on the boots, and then using a clean water solution on the boots too to rinse the extra bleach off after the germs are killed. And then another way we protect the ponds, you can see on my hand there, I have a glove on. All the children and all the scientists and everyone who was helping, we were wearing gloves to protect the spread of germs from us especially to the amphibians, because their skin is semi-permeable. We want to make sure nothing on us is getting into their habitat as well. So you'll see in a lot of the pictures we're all at least wearing one glove so that we can handle the amphibians safely. So of course, the next thing is, you know, okay, so it's not just me out there in these ponds with these kids. I did have a lot of help. I had my first community partner, the PARS representatives. There was one from Huntington County and one from Blair County that both agreed to come to some of the pond visits with us. And then also another community partner I partnered up with was the Huntington Conservation District. And the girl in the middle is a watershed specialist for the Conservation District. So her and then another person from the Conservation District who isn't in this picture, they also came to a lot of the pond visits. So at every pond visit, I always had either a Conservation District rep or a PARS rep or both, or sometimes, you know, all four of them would come to the pond depending on their schedule. So again, you know, picking those dates at the beginning of the project way back in January, February, and getting everybody's schedule squared away, that was all behind the scenes work that happened before the children even arrived on site. So, you know, if you're thinking of doing a project, you know, I mean, the pond trips were from March to mid-April, but really a lot of the hard work that was going on before the trips even started was starting 2 or 3 months ahead of time, you know. So also another person who was helping me with this was my partner for Juniata Valley After School Science Club. This is Kathy Kenny. She works at Juniata Valley Elementary with me, and her and I are co-advisors for the Science Club at Juniata Valley, the after school program. So she also was able to come to a few of the visits and give her assistance too with the children in the ponds. And again, like I mentioned, these pond visits, I invited literally every child in the school, in a school of about 400 kids, grades K through 6, and I had 47 kids sign up. So I thought that was really great, you know, that 50 kids out of about 400 signed up for the project, and I was able to accommodate them all with different trips in the van and adding those extra trips so that everyone would have a seat and be able to go. So you're probably wondering, okay, so we're out there wandering around in these ponds, what did we find? So first of all, like I said, the pond trips took place from beginning of March to mid-April. We chose those dates because of everyone's schedule, because of the van schedule, and also because of when we were going to see the most action in the ponds. We didn't want to show up with 6 kids at a pond and there not be much going on. And that's why I included the plants, because I figured even if there's only a few macroinvertebrates and no amphibians yet, we're at least going to see plants emerging like the skunk cabbage here in this picture. So you can see, you know, even though it's still kind of chilly, we're kind of still wandering around, finding all kinds of neat things like louseworts and skunk cabbage in the ponds. And what was really cool is the Conservation District is working on a project near one of the ponds to put some native plants in so that we can help with that buffer there between the stream and the forest. So we've got all kinds of plants going in here. We were putting, it was just little sticks we were shoving in the ground, but these are actually growing really well down there now. So we actually helped to, on that project with putting some plants in for the Conservation District. So you can see here, I just compiled the data when I did the Family Science Night. I hung these posters around the presentation that I was doing there, just to kind of show parents, you know, and to show children, you know, the long-reaching data collection from our project. So this is the one on plants. You can see I listed our other plants that we weren't focusing on, but we still listed all of them there so that the children could see what it looked like for the month-and-a-half span that we were working in the ponds for. So also, you know, we were looking for the macroinvertebrates, and this is kind of what I was envisioning when I was doing the dip netting up in Churchill. So you can see, you know, we're wandering all over the place, dipping our nets everywhere, trying to find any types of creatures we can, rolling over logs in the ponds and beside the ponds, just trying to find those macroinvertebrates. You see us using a lot of nets here. Some of the nets were provided by PARS. I had some already, and then I had purchased some with some Science Club money, just those little green nets from Walmart, just so that every child would at least have a net, because everybody wants to catch the salamander or frog once we start hearing them croaking. What was really cool is, you know, with the kids being different ages, grades K through 6, we did see a lot of assistance between different ages of children, you know, bigger kids taking the little kids under their wing, you know, kids of the same age working together to figure out some problems that needed solved. For example, you can see here the girls kept finding that water was getting down to their boots were getting swamped. So instead of dumping the water out on the road, they decided that they needed to dump the water out into their nets because they didn't want to kill any of the macroinvertebrates. So, you know, just respecting those animals' environment that we were working in while we were trying to find the animals. So not going helter-skelter crazy through the ponds, but, you know, trying to respect the environment that we were visiting. And we did have a lot of success. Macroinvertebrates, you know, they're everywhere. You're usually going to find them almost year-round in ponds and streams. So we were very lucky, you know, even at trips where we didn't see as many frogs or salamanders or turtles, we still were finding lots of macroinvertebrates. And we even found one I had never seen before, the water scorpion, which was pretty exciting for me because I've never seen that before. And the watershed specialist had never seen one before either. Unfortunately, she wasn't on that trip, but she did get to see the photographs. So she was really excited about the fact that those ones are in Huntington County. So here's our chart for macroinvertebrates. You can see pretty good spread over the 2 months. And then, of course, looking for eggs. Egg masses were starting as early as the beginning of March. And here we have a wonderful egg mass that we found at night as it was starting to get dark. And that was a frog egg mass. And here we have toads. These ones are in long strings. This was later in the season, so this is mid-April. This is about near the end of the project. And some of these eggs, I mean, we were just pulling up so many because in mid-April, it was really starting to get really active. And of course, the adult frogs, like the spring peepers. And, again, you're seeing the gloves, seeing the respect by the children. They really did a great job. Doing a little presentation before we went out into the field with them, I think that was a really smart way to do it. I thought about that and did it that way. I didn't want to bore the kids. I knew they'd be hopping to get in that van and to get to the ponds to find these animals. But I didn't want to damage the environment, because some of these ponds, there's a lot of, one of the ponds in particular, there was 3 different species of concern from Pennsylvania in it. So, we don't want to be damaging the environment and hurting the organisms. We found wood frogs also, and then bullfrogs and green frogs too. And then here you can really see how the frogs and toads really come out in April. March, they're just getting started, wood frogs and spring peepers, a little bit of bullfrogs. And then in April, we literally saw every type of frog that we were focusing on with our list of 6. So it's pretty exciting that we were able to find them all. What's really neat is the kids didn't get to see every pond visit, of course. Each child only got to go to 2 ponds for 1 evening's work. So that's why I decided that the family science night would be a great way to bring it all together so that they could see the whole project and what had been going on when they weren't out in the field with me. So we also were looking for salamander eggs and salamanders and newts. So you can see here the salamander eggs have a envelope around the whole egg mass and then single envelopes around each egg. So that's how you can tell the difference between some salamander egg masses and frog and toad egg masses. And of course, we were finding the larvae too, the baby salamanders. You can see they look a little different than tadpoles, baby frogs and toads. They look a little different. Here's some more egg masses we were finding pretty early in the season. And then the actual adult salamanders. A lot of the time we'd find those under logs near the ponds. Like I said, at one of the ponds, it was just great because we found 3 species of concern in Pennsylvania in one pond area. But then again, the importance, and the children and I talked about this, what if that pond was a clear-cut logging operation? What would happen to those 3 species of concern that lived in that pond area? So, just kind of trying to plant some seeds in the kids' heads about, appropriate use of resources and what happens when humans impact the land. So here we have using the identification guide, to identify the red-spotted newts, which we are finding in profusion pretty much everywhere. Another thing that was kind of situational, like us planting the red ochre trees and shrubs down in the one pond area with the conservation group. Also, one of the PARS representatives here, the man with the hat on, he was actually collecting samples from red-spotted newts to check on some funguses and diseases that we're having. So we're taking little tail samples here. So he's snipping with scissors just tiny, tiny bits of their tails so he can take those back to the lab. And then doing swabs with them, with a cotton swab, and then putting that inside a case. And then he was going to do some more research with some funguses and things and some viruses that the red-spotted newts were getting. So it was really cool. We actually got to be a part of those 2 side projects just situationally as part of this project. So that was pretty cool. Here's salamanders. We didn't see any marbled or redback salamanders, unfortunately, but we did see a lot of the other ones. It was really great. And then you can see the Valley and Ridge salamander on the others there, and the Jeffersons. Those are 2 of the species of concern, and the four-toed. That's another species of concern. We saw those both months. So it's really wonderful that we were able to see so many neat things out there. Also looking for turtles. This was the giant snapping turtle that we found that everyone in school was talking about for weeks after we found it. It was so exciting. You could see the size of it. And of course, you know, again, a learning situation, you know, teaching children that, you know, we don't pick up a snapping turtle. This PARS representative is specially trained in picking up snapping turtles safely so that he doesn't lose his fingers. So, you know, he wanted to pick it up and show it to the children, and he basically dove into the water like Steve Irwin used to with the crocodiles, and got the snapping turtle and showed the children. So you could see their reactions to the snapping turtle. It was a pretty cool moment for the students. You can see the size of it there. I mean, it was a pretty big one. And I guess just in this picture here, you can see hanging from my waders, you see that little yellow tag there. That's actually a fishing license. I guess I should have said at the beginning, to be able to collect but still not even take home organisms from ponds and lakes and streams, you do need to have in Pennsylvania a Pennsylvania fishing license. And that's even just to look at them and touch them and take them out of the ponds and then put them back, not even taking them home. So again, you know, you might want to, when you're working out in the field, you know, you do have to look at regulations and laws and make sure, you know, that you're following the laws for your state or your area. And then we also found near the end of the season some baby turtles, which was pretty cool. Some baby painted turtles. So you can see here, didn't find tons of turtles. You know, probably in May, as it gets warmer, is when a lot of these other turtles would be starting to pop out. But we did get to see kind of the beginning of the turtle period of the year. And then we weren't looking for snakes, but we found some. So at one of the sites especially, there was a lot of garter snakes. And the children were very excited. We don't have our gloves on. This is a reptile, so it's probably okay to be touching it without gloves on. Their skin isn't as sensitive as the amphibians. And this we actually found as we were walking to a site, so we weren't gloved up yet. And this is after we were at the site, we found another one. And this little girl loved this garter snake. It was crawling in and out of her boot, and she was so excited about it. So I still talk to her about it today. You can see here the signs of animals and other things that we found, garter snakes, and then signs of mammals and birds and fish, you know, because these ponds don't just belong to the amphibians and reptiles. You know, there's lots of ecosystem interactions going on in these ponds. So, you know, all this data we collected, you know, in Churchill, obviously the scientists are studying something there. They're on a science project. I still wanted us to be able to share our data just like real scientists do. So again, helping out with PARS. We have an account on PARS. We uploaded our photographs, we uploaded our data, we uploaded all of our findings that we found. And you can check them out on that website. And also their information page was, again, one of the handouts that was emailed to you. So I don't know what state you're in, but I'm sure there's something in your state, the Fish Commission, the Game Commission. There's always something where people are doing citizen science and collecting data online. You're doing birds, eBird is a great website. So there's all kinds of things out there nowadays. There's a plant phenology network, you know, there's Nature's Notebook. There's all kinds of neat places where citizen science is going on. So this is the site that we use because of our PARS partners and because now our data goes to their survey, you know, to help them make decisions about what's going on in Pennsylvania with the amphibians and reptiles here in our state. I thought something really important, you know, with the science was to help the kids respond with art. You know, this wasn't something that happened during the school day, so we weren't doing writing and we weren't doing reading and we weren't doing books. But I thought a good way in an after-school setting to have children respond would be to do some art. So you can see here this little guy and his orb snail. He was fascinated by the orb snails. He kept finding them at a certain site. So he really worked hard. This is a kindergarten student to make a painting of his orb snail. Here's some water striders. This is, I think, a 4th or 3rd grader. Of course, the garter snake, very exciting. Everybody loved the garter snake. And then here we have kind of a pond scene with the spotted salamander. And this was a 6th grader. He was very meticulous in his art. He did not get finished on the night of the event. So I invited him during some break times that he had during the day for him to come down to my kindergarten classroom and to work on his art some more. And he came to my room for about 3 days for half an hour each to complete his painting of the baby painted turtle that he had found. So pretty cool that he was so interested in completing his response to the pond project. We also responded with music because I think it's important to incorporate the arts into science. I think it helps the kids connect better to the science that they're doing so that it's not just random data they're collecting. So these lyrics here were written by Jeff Rhodes, our music teacher at our school district. He's worked with me on different science club projects before. We had a night science where he wrote songs about night animals, and he writes all these lyrics himself and then puts them to sometimes well-known songs, and sometimes he writes his own melody. These two lyrics here, these are Go to the Song on Top of Spaghetti. And then what we did is at the family science night, I had the children perform the song for their families. So the verses in the song are about a bullfrog who kind of finds a snapping turtle, and then he gets eaten by the snapping turtle. And it's a really funny song. It's one of those comedy songs that our teacher, Jeff Rhodes, likes to write. So you can see the ending there is that you have to keep an eye on your bullfrog, because otherwise, when you glance at the sun and get blinded by the sun rays, the snapping turtle will bite your bullfrog and eat it. But it was really neat because a lot of his lyrics had to do with predator-prey interactions, ecosystem interactions. So it was neat because it was a good song for the children to sing because it was funny, but also kind of reminding them, you know, that everything in that environment is interacting. And then we did have the family science night so that families could see the art, so families could see the children perform their song. What I did at family science night, this was a big project. The whole time we were in the ponds, I didn't really have this mapped out completely, but I knew I wanted to do something to show the parents of these children, then any families whose children maybe couldn't make it to a pond visit, who wanted to learn more about ponds, to come to a fun science night. So I decided in the beginning that I was definitely going to do something, but I wasn't quite sure what it was going to look like. Then about mid-March, I thought, well, I better get started here. So I started inviting lots of friends that I knew from different organizations, you know, like the Game Commission, you know, Vernal Pool Registry, our Audubon Society, of course, our PARS partners and our conservation partners, and talking to the PTO and our high school sustainability club, and, you know, trying to get all these people on board so that they could at least come and maybe either give a talk or have a booth. And I decided what I was going to do then when I thought about it. I did a little presentation for the parents and families just so they could see kind of photographs of their children working in the ponds, but also to share the posters of our data, and also to show the children, you know, like with the posters of the data, how the ponds came more and more alive in April. And that really was attended pretty well. Out of the 47 kids who participated, there was about 39 whose families came. And then some additional families came whose children didn't participate in the pond, because I invited all the children in the school to come to family science night. So total, there was about 50 families there, which was great. And you can see at the different interactive booths, I did a little presentation. We sang the song. I had the children's art hanging everywhere. And then we had interactive booths. So you can see here the Conservation District booth, where they're showing the macroinvertebrates, and our PARS partners showing live amphibians and reptiles and identification guides to the children. And then we have our Audubon Society, who brought in a booth about wetland birds. And then having the Department of Forestry come in, and he brought in this really neat thing where there's creatures in there, little salamanders and worms and different things, and the children could kind of go on their own little mini beside the pond hunt there. And here we have the Vernal Pool Registry. This is an organization that all the vernal pools in the whole state of Pennsylvania can actually be registered and documented, their locations and what life is there. So she came and provided some posters to children. And then having our local environmental center come and bring live snakes. And having our Pennsylvania Native Plant Society come and talk about wetland plants. And each child that visited the booth got to plant a wetland plant seed and take it home with them. And then having someone from a local state park come in, Canoe Creek State Park come in, and she brought some pond water and some different magnifiers and models of animals to look at. And then this is our high school sustainability club advisor. She had brought a watershed quality type thing where you could use this model to pour water onto it and then see where polluted water would go if pollution had leaked out of a certain point source. Having the Game Commission come and bringing mammals to show the mammals that you might find around ponds. And then this is our PTO, and they designed all these pond snacks. As you can see, the pond slime and different things that they made, these creative pond foods for the children to enjoy. So in a nutshell, that was my project. And you know, it started small and it kind of hairballed into this giant thing that kept me working night after night at school really late. But I think it's really important to make connections from your work when you go out on a science expedition. you know, to help students learn to work as citizen scientists in the field. And, you know, even if you don't go on an expedition or you're just interested in science, like, I think it's really neat to show students that they can actually be citizen scientists in the field. You know, you talk to students at the beginning of one of these projects and you ask them if they're scientists, and a lot of them say they're not. And then after one of these projects is over, they really feel that they're scientists. You know, and I feel that every person is a scientist. You know, you're wondering about things around you and exploring the world around you. So that makes you a citizen scientist. And I always explain to the kids, like, okay, we're not getting paid for this. That means we're citizen scientists, but we're still scientists. And they really seem to feel that they're scientists after doing a project like this. So what's next for me is I'm working on a new project now at the school. You can see here our open top warming chamber and our ambient temperature plots. This was from last spring also. And I'm working with Dr. Eric Post from UC Davis on some different climate change experiments with plants now for plant phenology. So, and then I'm going to bring all those projects together this coming spring at another family science night dealing with plants and phenology and seasonal changes. We had done a basil plot crop production study in the spring, and then that continued till the summer, and then it continued to this fall when we finally harvested all the basil. So that's the next thing that I'm going to be working on. And if you need help getting your project started, or maybe you have a question about projects, maybe I wasn't specific enough about how I got this thing going or what I specifically did to map out the ponds, or maybe you're interested in studying birds or some other type of ecosystem, you know, maybe I can help you out. This is a photo of this binder is all the data that was collected in Churchill when I went up to Churchill. And I mean, it was amazing, this giant binder of data. So, you know, maybe I can help you out. I've been on 6 expeditions now, some with Earthwatch and some with other organizations. So hopefully if you have any questions, here's my contact information. And if I can help you out more, just let me know. And if you have any questions now, I'd be, have to get off of this thing. And then I can take your, I think it's typed questions, and see if you have any questions now.
Great, thank you so much, Kristin. And yeah, as you said, if you do have any questions, just type them in the chat box for all panelists, and we'll go from there. So the first question, Kristin, is looking back, what would you do differently with your pond project?
So I think, yeah, I think, can everyone hear me if I talk, or do I have to type it in?
Nope, we can hear you. You're perfect.
Okay, thank you. So thinking about the pond project and thinking about what I'm going to do this year with my plants and my phenology projects, I definitely think looking back, I would have started a lot earlier. Of course, you know, we started driving around and looking at ponds in January. Thinking back in retrospect, I probably should have started in September. Because there were some really late nights where I was frantically getting things put together before I had 6 kids showing up, because of, you know, oh wow, I started a couple months ago, but still wasn't enough time for me. So that might be something that I would have changed for next time.
Great, thank you. And you've got a comment saying, great job sharing your hard work.
This is from my principal, so thank you, Mrs. Quinner. I appreciate you attending, and I'm glad you're proud to have me at Indiana Valley. I really appreciate it.
Okay, we'll give everyone 1 more minute if anyone else has any questions to type in for the group. And if not, as Kristin mentioned, you're also welcome to reach out to her individually. You're getting some more comments in here.
Thank you. Yes, I mean, you know, I see here, you know, somebody commented about wishing they had opportunities in elementary school like this. Yeah, I mean, I think, I don't know how many attendees are elementary educators, but, you know, I think it's really important to incorporate real science into the curriculum as much as possible. Like doing science activities is great, but if you can incorporate real science research into your curriculum in any way, I think it's really important for students to see, you know, that it's real science. It's not just using baking soda and vinegar to make a muffin, which is really cool and really fun, and it should be done. but also incorporating some real science research into your children's lessons too. I think that can be really meaningful in students' lives.
And another question here is, did your school fund the expeditions? Buses are hard to pay for in Gabriella's area for after-school programming, so just wondering how you were able to have the van and fund the project.
Okay, right, yeah. So funding for after-school programming is what the question is?
Yeah, this teacher has trouble with her after-school programming fund, so I was wondering how you about it.
Yeah, that can be a real challenge. So to answer that teacher's question, in a nutshell, my friend, Mr. Paul Gregory, was our science club advisor for many, many years, like since I was in elementary school at a different school. And when I started at Juniata Valley, I was like, who's this crazy science man running around with chemistry boots on and stuff all the time? I want to do stuff with this teacher. So I introduced myself and started volunteering at his after-school science club. He had a contract for to get paid. And then from there, I really enjoyed doing the after-school program. And so he and I, and this is where the question for the teacher, this might really help you. He and I went to the school board. I had volunteered with his group for an entire 2 school years. We took photographs of all the work that we did together and made a video. And we showed the school board why 2 people needed to help with science club after school if the science club is going to continue with all of the grades K through 6. So it's not really a job for one person, grades K through 6. So that was kind of how I got my contract with science club about 6 years ago. So, I suggest, getting your school board on board for your programs. Maybe that's one way to try to get something. A lot of the time it's easier to get funding for after school stuff is if you do a lot of work first and then show that you did all this work and how useful it'll be to have that for children. Sometimes that's a way to try. I know it's really hard. And sometimes if you do something for free for a while, sometimes it's harder to get money for it. So, just do your best. Materials wise, I spent my own money on some of those nets that I bought for the project, knowing that I'll use them again for another project, no big deal. But the van and everything, that's all paid for by the school, the gas for the van. I bought the snacks for the kids with my own money, but my contract was paid by the school too as an after school curriculum. So do your best. There's also grants you can apply for, lots of different grants out there nowadays. So maybe look into that. Different states do different grants.
Great, thanks. And we have Elaine here saying, can you travel to Nicholson, Pennsylvania and check out their creek?
I can do that, Annie Lane. Yes, we'll have to maybe we can do a creek study the next time I visit Nicholson. That would be really cool. Maybe we can go to Francis Slocum and check out their lake and see what's in there.
And then it looks like this is the last one from Diane saying, I think this would be a great tool to educate adults in the community. I wonder if it could go beyond this into education and programs in state park systems using your plan.
Yeah, definitely. When I had the Canoe Creek lady who came to our family science night, she was really excited about saying, they could do some events at Canoe Creek State Park incorporating a community, pond, or I guess for them it's lake, community lake and creek project where they could have macroorganisms for parents and then of course their children they would hopefully bring with them. But sometimes at those state parks, it's just adults coming to the events and that's okay. But yeah, I definitely think a good model for state parks is to try to get adults and children interested and in the field doing things.
And I know I was actually recently up on our Earthwatch expedition in Acadia National Park in Maine, and there they do some education programming such as bio blitzes, where adults and students of sort of all ages are there gathering data for a day in the state park. So I would look up what local bio blitzes are maybe happening in your area.
Yeah, great suggestion. Yeah, I've been on bio blitzes and they really are wonderful. They're really neat, and they're not usually geared towards children. They're usually geared towards adults because you are doing a lot of collection with science. 24 hours usually.
Yeah, definitely. And it looks like one other quick thing following up on Gabriella's question about the funding, a recommendation to try local businesses and see if they'd be willing to donate supplies and food and that sort of thing as well. If you kind of come to them with your plan and see if they'd be willing to pitch in, whatever.
Yeah.
Right. That's a great recommendation.
I know Walmart's will do, Walmart's are pretty generous about donating small amounts of money, usually just to go to the customer service desk and ask them about their school donation program. And then there's just a form you fill out and write what materials you want them to donate. They're usually really good about quickly donating. So take a look even at your local Walmart, you know, to get at least a small amount of money or supplies. And also parents, you know, I had our PTO come and supply the snacks at the family science site. You know, sometimes PTOs and that kind of thing can help out.
Great. Well, it looks like that's it. So thank you so much, Kristin, for your time and your wonderful presentation. I hope that inspires some of you all to go out and start your own projects. And as Kristin said, reach out if you have any questions as you're trying to get it started. She did a really great job, and it looks like more to come this year with the project. So great role model as you're getting that started in your own school. Thanks, Kristin.
Yeah, thank you everyone for coming. I appreciate you coming.
Have a great evening, everyone. Talk to you all soon.
Thank you.